Hello, everyone, and welcome to today's webinar, GenScript's Response to the SARS-CoV-2 Pandemic, Innovations in Diagnostic Solutions. This webinar is part of the 12th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics Virtual Event Series 2024. I am Israel Gonzalez, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by GenScript. For more information about GenScript, visit www.genscript.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation by submitting them into the Ask a Question box. We'll answer as many questions as we have time for at the end of the presentation. Also, if you have any trouble seeing or hearing the presentation, you can click the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's go ahead and get started. I now present today's speaker, Amanda Grimm, Senior Segment Marketing Manager, Antibody Drug Discovery at GenScript USA Incorporated. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Amanda, you may now begin your presentation.
Thank you so much for the introduction. Let's go ahead and get started then. So here I just have an overview of what we'll get into today. I'll provide some information about GenScript. I have a couple of slides about precision medicine and in vitro diagnostics. We'll talk about GenScript's journey into diagnostics. I'll talk about solutions, some customer highlights, and what it looks like to partner with GenScript. So we'll start with some information, about GenScript. For those of you not familiar with GenScript, we have over 20 years of development experience. So long-time industry experience, most notable for our gene synthesis and plasmid services. We're always at the forefront to provide the newest technology to improve your methods and output. Our CRO and CDMO foundation provides us with a range of experience, having multiple partners in the therapeutic space and working with them to bring their antibody drug, their cell or gene therapy, or vaccine to market. And then because of COVID, which we'll get into here shortly, we were able to offer the first of its kind neutralizing antibody detection kit across multiple countries and regulatory bodies, adding to our regulatory experience. So here is a little bit of the history and milestones of the GenScript group. We were founded in 2002. Since that time, we've added some additional subsidiaries like Legend Biotech, ProBio, which is our CDMO, and we continue to grow and build out. Here's just a little bit more information about individual subsidiaries. So say I'm with the Life Science CRO, and then we have our CDMO ProBio. Legend Biotech is our cell therapy unit, and then we have Bestzyme, that is our synthetic biology company. So as I mentioned, I have a couple slides to touch on regarding precision medicine and in vitro diagnostics. And that's really to highlight the importance of companion diagnostics, which are going to be those diagnostics that support new drug and therapeutic development. This applies to many disease areas like cancer, infectious disease, and can include a different type of antibody, can include different types of diagnostic tests, including immunoassays that involve antibodies, and also something like next-gen sequencing. So these, in the best case scenario, they should be developed in tandem for that aligned performance so that they meet regulatory requirements. And it's ultimately going to involve some education and training for the physicians as well as lab workers in order to understand the benefits of both and how they work together, and ultimately getting labs ready to provide that testing and understand where the testing fits in with other standard testing that may already be performed for that particular disease. And then a nice example of a companion diagnostic is this one for acute myeloid leukemia, which of course is a cancer. We have Abbott's test, the real-time IDH1. This is for the detection of the IDH1 R132 mutations in the blood or bone marrow. And this test ties directly to Tibsovo in that treatment, since it's specific to patients with AML and are positive for this mutation. Next, I want to take you into GenScript's journey with diagnostics. And this, I think, is interesting because GenScript isn't necessarily a diagnostics company, although we offer the services and products that do contribute to making diagnostics. The COVID-19 pandemic really opened this opportunity since many companies in our industry were pivoting to offer solutions to help. This is just a brief timeline to remind us a little of the events as it relates to GenScript. Like say we had the emergence in late 2019, and then WHO declares a pandemic in March 2020. And by the end of 2020, we really see many treatments available, and our first vaccine also available through that FDA EUA. So while a vaccine is a preventative measure, the treatments were really evaluated for patients who were experiencing degrees of severity for the disease. GenScript's cPass was initially on the market as an RUO or lab-developed test, and then it did receive the first FDA EUA as a clinical diagnostic in November 2021. And then, as we all know, May of last year, the pandemic was declared no longer emergency stage by WHO, and then the US also followed with removing the public health emergency status. But that said, we do still have cPass available for clinical use, as well as the RUO version, which is this, referred to as this surrogate viral neutralization test. And these are used for many studies to further understand COVID. So some information to start into the insight of GenScript tests, like say our first assay to detect COVID neutralizing antibodies in human blood. Early in the pandemic, this would indicate presence, absence of those SARS-CoV-2 neutralizing antibodies. We later added a quantitative protocol in order to quantify those neutralizing antibodies. This assay was a collaboration with Dr. Lin-Fa Wang at Duke NUS in Singapore, whereby we were able to commercialize the kit based on his research to create a novel assay. We have the RUO version and we have cPass, which, like say, they're essentially the same test, just cPass was granted the EUA. And then the test has also received other regulatory authorizations in other countries like Europe, Brazil, and Singapore. The assay achievement really demonstrated GenScript's ability to create, manufacture, and market a diagnostic kit in the regulatory environment. So some more information about this assay from research to commercialization. This assay is a competitive ELISA that replicates the conventional virus neutralization test into what is known as surrogate technology. It's based on the knowledge that the virus binds its RBD to our cell's ACE2 receptor, and that neutralizing antibodies, if present, are going to block this action. Thus, the assay utilizes recombinant ACE2 antigen, which is coated on a microtiter plate, and then uses secondary reagents of recombinant RBD that then compete with patient samples containing the target antibody if it's present. The kit materials listed over on the right were produced by GenScript and include the controls that are also antibody-based, like say the recombinant ACE2 antigen. We have the HRP conjugated recombinant RBD and the buffers. And then just some background for diagnostic significance. I've tried to highlight here with a few papers. This top publication is a publication from 2021 that provides a review of the information available at the time about neutralizing antibodies and what they mean for COVID infections from a clinical perspective, noting that traditionally and in some cases, neutralizing antibody titers could be linked with disease severity, while noting that the disease is dynamic and did have many missing puzzle pieces like combination effects of neutralizing antibody with cell-mediated immunity. Both cPass and the RUO versions provided and continue to provide insights into the immune response from natural infection and or vaccination in humans. It also has contributed to knowledge in the veterinary health industry, particularly used in this study to surveil white-tailed deer. Some studies provided more specific insights into groups of individuals that may have compromised immune systems, such as this publication on the left, looking at individuals that were immunocompromised due to solid organ transplant and looking at their immune response to vaccination. In addition to broad antibody and neutralizing antibody data, they also measured T-cell response. And the combination of this data was helpful when also additionally combined with other individual health data so that physicians could provide more specific health recommendations for their patients during this time. The second assay we commercialized was our qPCR kit to detect viral nucleic acid in patient samples. This achieved the CE mark in the EU early in the pandemic, and it was mostly available outside the US. This test was based on TaqMan qPCR technology. It was developed based on early and available coronavirus research at the time. So the primers and probes were based on the unique ORF1AB, nucleocapsid, the RDRP, and E genes. Customers essentially could choose to do a single or dual labeled testing. And then the output of this was a quantitative measurement using general qPCR instrumentation. So we were really able to take advantage of what we were already able to do for our customers and transfer that knowledge into actual diagnostics. So in this next section, I want to share our IVD development solutions. And here to start, just to kind of set the stage, we have a high-level overview of the development process where it starts with a disease of interest, identifying a target, designing and optimizing that assay, some validation and verification. And then once approved, it can be used for diagnostic purposes. So from the development workflow, we have 3 stages that we've identified where we can provide solutions, that being target identification, assay format and validation, and then assay production and quality control. GenScript's offerings align primarily with 2 IVD formats, the first being antibody or antigen detection, and the second being molecular detection. So starting with target identification in this stage for this purpose, it evaluates the target feasibility and whether it is possible to detect with the available technology and resources. Targets here include proteins, antibodies, or nucleic acids. Common examples include pathogen-produced protein epitopes, cytokines, enzymes, or measuring an antibody response. Target identification may involve a number of methodologies in order to confirm the target functionality related to the disease. And this specifically is subdivided further into 2 areas of analyses, gene analysis and protein analysis. So starting with gene analysis, like say we want to ensure the ability to evaluate those target genes and their functionality. So the Gene Titan 8 million oligosynthesis platform enables the efficient production of high quality oligonucleotides that can be DNA or RNA probes for target capture and detection. The NGS next-gen sequencing technology complements this process by providing comprehensive sequencing and analysis of target regions, allowing for that in-depth characterization and profiling. The amplicon-based approach involves PCR amplification of specific target regions, including primers designed to capture the desired genomic regions of interest. This method allows for targeted sequencing of specific regions, enabling the detection of genetic variant mutations or biomarkers associated with your disease of interest. And then the hybridization-based approach, this involves the capture and enrichment of target sequences using custom designed probes or baits. These probes are designed to specifically bind to the regions of interest in the genome, allowing for selective capture and subsequent sequencing of the targeted genomic regions. Continuing in the evaluation of target genes and functionality, we have a comprehensive range of ready-to-use PCR, qPCR reagents, and complementary custom services. The ready-to-use reagents for PCR, qPCR are going to be formulated to provide reliable amplification of target DNA or RNA sequences, resulting in sensitive and specific detection. Our oligo custom services, including primer and probe design, optimization, and synthesis, will ensure the successful identification of specific targets. The primers and probes can be provided to you in a tube or even a plate format if you are conducting high throughput testing. Antibodies provide an integral role in development where they can function in target characterization, antigen capture, or antigen detection. Our catalog products division offers a wide range of antibodies that can be purchased ready to use. Our range includes anti-VHH, epitope tag, anti-idiotype, and immunodetection. And then we also have a large catalog of IVD antibodies that includes antibodies and antibody pairs for conditions like bone metabolism, fertility, and pregnancy, and some that are tumor specific. These antibodies have been fully characterized and they indicate specific use applications that they've been tested for. So some examples of that would be ELISA or flow cytometry. GenScript also has a range of antibody services that can be fully customized, whether that's through antigen optimization or incorporating our proprietary immunization technology. The options include polyclonal, traditional rodent hybridoma, and of course our monorab. The new TurboMab platform is mouse-based, similar to our hybridoma, but it incorporates positive cell enrichment with the option to include B-cell screening and cloning, similar to what you would get with the monorab. And this will further enhance sensitive antibodies as a result. The monorab, I should note, is frequently requested platform. The rabbit immune system offers a significant advantage with its unique immune system in order to achieve highly sensitive and specific antibodies with little to no crossover. So the products division offers a comprehensive suite of instruments and reagents for Western blot automation to support protein analysis and characterization. Once expressed and purified, your proteins can be checked for size confirmation, and denatured or native conformation reactions can be done via that downstream testing. The precast gels are available in various formats and percentages, so you can choose whichever is most suitable for your specific experiment. In addition to gels, we offer equipment to support each stage of that Western blot process. So that includes transfer systems, blotting, as well as staining. To complement the work With gels and equipment, we also provide an extensive collection of catalog elite antibodies to further assist with that protein analysis. Our custom peptide synthesis generates peptides corresponding to your specific target sequence of interest. These peptides can be used for epitope mapping, antibody validation, target antigens, or as antigens for antibody production. The peptide service also includes peptide design, modification, and labeling options to meet specific project needs. In addition to the types of peptides here, we can also offer libraries, modification, as well as labeling. And then our protein expression service covers multiple expression platforms to accommodate the diverse needs and to optimize and scale production. Our mammalian expression platform offers the advantage of post-translational modifications and native folding, making it suitable for the production of complex proteins and antibodies that require proper folding and glycosylation. The system ensures the generation of biologically active proteins that closely resemble their native counterparts. We have HEK and CHO lines, and I'll cover the TurboCHO platform more in depth in our next slide. The insect expression platform offers an alternative for the production of challenging proteins that require proper folding and post-translational modifications. This is an alternative method in order to produce recombinant proteins with a high yield and functionality. For more simple protein expression needs, of course, we have E. coli that provides a cost-effective as well as a rapid solution. It's going to be ideal for the expression of soluble proteins, protein fragments, or small-scale production of antibodies for initial screening and characterization. So our CHO expression is our proprietary TurboCHO platform that offers a scalable platform for recombinant antibody and proteins. So as the development process scales, we can scale the expression and production along the way. The high throughput in the discovery stage is a transient expression that provides the fastest turnaround at 5 business days. Then we would move to express as the protein amount needs increase. It's also transient, but can also support many different antibody formats. And then to high performance and stable pools in order to support the later stage where large stable quantities are needed for those clinical studies and commercialization. As an alternative to Western, we have our ELISA development service that provides a robust platform for screening and identifying potential targets or even as prototypes to their assay format. In addition, custom ELISAs are also valuable to assess the binding affinity, specificity, and activity of antibodies and proteins. So now let's take a look at the next phase of development, assay format and validation. Common assay formats include ELISA, lateral flow, PCR, qPCR, and cell or tissue-based assays. Assay development considerations include relevant controls, the run conditions, and the reagents necessary to perform the test. Once the design is complete, conditions are optimized further to ensure the assay is sensitive, specific, and reproducible. So in this next section, I'll divide out between the antibody and antigen detection and then the molecular detection development. So starting with antibody antigen detection assays like ELISAs and rapid tests. Let's say you have antibodies or an assay that uses an antibody. The precision mutant libraries and mutation services are going to be great tools to facilitate development of more accurate and sensitive diagnostic assays. The precision mutant libraries consist of a collection of systematically designed mutants that enable you to explore the functional significance of specific mutations in their target proteins. Our mutation services allow for the generation of custom designed mutants, providing flexibility in studying and optimizing antibodies. Mutation options include the site-directed saturation scanning and combinatorial mutagenesis. Performing the optimization and validation testing is going to require large volumes of antibody to make sure there's enough for your fine-tuning and that optimal performance to get accuracy and the reliability without having to constantly make more batches. It's also an important part of the scale-up process for your assay materials. And we can offer both in vivo ascites or in vitro using roller bottle scale-up of hybridoma production. I did break this section into 2 slides just to simplify the information. So here we see the ascites production workflow that's ultimately going to give you 2 to 5 mils of 1 to 10 mg per mil per mouse. And then here we're looking at the hybridoma workflow where we ultimately can go one of 2 routes. The first being hybridoma expression across the top yielding 15 to 50 milligrams per mil starting at a single liter production volume. And then the second is sequencing that antibody to generate a recombinant antibody that can help to increase your yields, generating over 100 mg per liter in volumes ranging from 100 mils to 200 liters. Antibodies or proteins may require labeling with a fluorescent dye or enzyme like horseradish peroxidase. So when you think of assays like ELISAs that use a substrate and enzymatic reaction to generate a color change, or even flow cytometry where you would be separating cells based on a fluorescent marker emission. These are options that are part of our antibody reagent modification and purification services. As mentioned earlier in the target identification stage, we do offer a catalog of ready-to-use IVD antibodies that address many commonly investigated health conditions and diseases that include bone metabolism, kidney markers, diabetes, and Alzheimer's. These antibodies are produced with high-quality standards and have shown superior lot-to-lot consistency. These antibodies are highly characterized, so we know their isotype, we know their working format, whether that's immunofluorescence, ELISA, etc. And also we can recommend antibody pairs. These are ready to use and can be purchased in large quantities. Here we'll transition over to assay design and optimization for molecular detection assays. And I'll mention here, CRISPR is being widely used in many applications, and diagnostics is one seeing many benefits. We have Sherlock, which is a company that was the first to receive FDA EUA for their CRISPR-based COVID lateral flow assay. This is a diagram from the Kaminsky publication outlining some of the different preamplification pathways and different associated Cas enzymes used to develop different types of diagnostics. GenScript does have ready-to-use Cas nucleases that cover each of the Cas9, 12A, and 13A, as shown in the chart over here. We also offer customized single guide RNA and CRISPR enzyme services as well. For PCR and qPCR materials, these can also be purchased in larger quantities for optimizing your nucleic acid detection assay. We do offer a number of catalog product reagents of interest, including those enzymes, the dNTPs, and plasmid vectors. Our oligo synthesis service provides primer and probe product with low contamination, highly precise and sensitive, consistent labeling, and a very stable product. The oligos are important, whether they are part of the reaction or if they're functioning as a positive or negative control. All right, moving on. The validation of a diagnostic assay adheres to the processes outlined by local regulations. In the United States, the 510(k) trial and the de novo established by the FDA involves collecting sample data for analysis. This analysis evaluates the precision, accuracy, reportable range, reference range, analytical sensitivity, and specificity of that assay. So to maintain accuracy and reliability over time, quality control procedures are implemented early on and carried through to commercialization. By this point, your materials will be produced according to internal development, where those materials must meet set qualification standards while produced according to set quality measures. I do want to preface this section that ISO production may be a customer request of the product we provide them. And we can also elevate the quality standard of other materials not listed here. We do want to assure that the raw materials used in your diagnostic assays are of the highest quality, meeting regulatory requirements and contributing to the accuracy and reliability of the final diagnostic product. So again, we'll start this section looking at antibody and antigen detection assays. Again, examples, ELISA and rapid tests. And for high quality protein or antibody production, we have the high performance option from the TurboCHO platform previously mentioned. It's a key option for expression of high amounts of protein or antibody that will give you from 500 milligrams to kilogram levels. This can be done in as little as 6 weeks in order to express recombinant proteins or antibodies. And then our QC testing standards for this include SDS-PAGE and endotoxin control. We do have additional options like FACS or SPR that customers can request for their projects. We also have GenScript's large-scale antibody manufacturing services. I did cover this with the ascites and hybridoma. So this offers scale-up production for monoclonal and polyclonal antibodies from milligram to gram levels. This works for both the in vitro antibody scale-up hybridoma technologies, such as roller bottle cell cultures, and then also the in vivo ascites production for hybridomas that may perform poorly in culture. Each is available with mycoplasma testing, mycoplasma removal, and the clonality services for the highest quality of your antibody production. Our total quality management platform is used for the peptide synthesis and is ISO 9001 certified. This ensures that custom peptides are triple checked for quality using both mass spec and HPLC after each step during the peptide purification and quality control procedures. As the final step in this platform, we perform additional quality assurance procedures for every custom peptide to further guarantee that delivery of high quality peptides for you. Now we'll take and transition over to the molecular detection. So as mentioned previously, CRISPR is a new technology being incorporated into diagnostics. We have GMP CRISPR capabilities already in action for our therapeutics materials, so we are able to take advantage of this to be able to adapt with the QMS level production you need for your assay. Oligosynthesis is a key service for primer and probe development, even next-gen sequencing, which I didn't talk too much in this presentation. We were able to provide this at ISO quality level, and then the deliverables for this include C of A and MS reports. In this next section, I wanted to take a moment to highlight a couple of our customers and their work as they've been published in peer-reviewed journals. This first customer highlight, I think, is an interesting study for chickens. Chickens and other poultry have a number of diseases that they're routinely screened for and also vaccinated for, and then monitored with regular testing. This paper outlines that interferons are not well characterized in chickens versus humans. And they cite this 2018 paper showing some supportive evidence that interferon kappa actually has potential antiviral activity. So they want to look to develop an assay to allow further research into the innate immunity mediated by chicken interferon kappa. Their study was successful in showing native detection, and the GenScript support they utilized was protein expression for recombinant antigen made in E. coli. And then they also utilized their antibody development platform to generate mouse monoclonal antibodies. In the second customer highlight, they identified a need for better cancer screening in older dogs. They found a need for a test that also is relatively easy to access with low cost point for owners. And the assay is based on knowledge that thymidine kinase 1 was successful in humans as a biomarker. The results of this were promising, and then it showed that canine TK1 could be a blood-based biomarker and could be used to monitor treatment in addition to that initial diagnosis. So for this, they utilized GenScript's antibody platform in order to generate 2 mouse hybridoma monoclonals using the predetermined TK1 peptide sequences. And then here I want to share that 3 of the 23 awardees from our newly launched Life Science Research Grant Program were actually for diagnostics projects, those being BioStone Animal Health, Mom Diagnostics, and then some work being done at the University of Pennsylvania. I was hoping by the time that I gave this talk, I'd be able to provide more specifics about the work each are doing. So definitely follow us on social media and email for these updates. And then this last section, I want to share some info just as it relates to partnering with us for work that you may need some help with. Here again, some spotlight data as of the end of June 2022. So I know we have definitely exceeded these numbers, but note that we've been cited in more than 74,000 peer-reviewed journals, and as indicated by the graph below, this number continues to grow. This is a summary of what it looks like to work with us. It can be as simple as some plasmid work, or if you wanted a more integrated flow, that's possible as well. Our teams love to provide support, and we hear that feedback often where customers are happy with the product deliveries, and they never have to question what's happening since our project managers are amazing to keep you up to date. And with that, I thank you for your time and your attendance, and I'll welcome any questions.
Awesome. Thank you so much, Amanda, for your informative presentation. We'll now start the Q&A portion of the webinar. So if you have any questions that you'd like to ask, please do so now. We already have some coming in. Our first question is, can you also help me identify antibody pairs during antibody screening?
Yes, we definitely can. We would incorporate that into the antibody criteria of the project during the screening in order to make sure your antibodies will work for your intended application.
Great, thank you. Our next question is, if the antibody specificity is not high enough, can you optimize it further?
Yeah, we can do that through a couple of different methods. We would further optimize probably through codon optimization, but also an important service to do this would be the precision mutant libraries, since by making multiple mutations, those antibodies could then be further tested for that increased specificity that you're looking for.
Great, thank you so much. Our third question is, what kind of labels can you add to the antibody?
Yeah, these are great questions. I did mention very quickly that we do fluorescent labels and also that HRP conjugation. But to specify further, we do also conjugate biotin. And then we have a pretty wide range of fluorophores that include like FITC, PE, many of the Alexas, and even APC.
Perfect, thank you. We have another one here. Can you tell me more about the research grant and how can I apply?
Yes, I'm so glad someone asked because we're really excited to offer this. In short, we're awarding applicants up to 1,000K in services to conduct your project. The projects span cell and gene therapy, antibody drug development, vaccines, and even diagnostics, as I mentioned. You can find this link on our homepage as one of the rotating promotions and also under resources. This page is going to have more information on the specific timelines and the application process. I know our Q2 application deadline is coming up very quickly, June 15th, so definitely check it out.
Great, thank you. And it looks like we have time for just one last question. And is this, I have a difficult antigen. What options for producing the antigen and immunization do you have?
Yeah, okay. We do have multiple options. We could start first with looking at the sequence, whether it requires some optimization that could be done, and also evaluate the expression method because maybe it needs some post-translational modifications, or maybe there's a folding issue going on. As for antigens for immunization, we can really work with anything. The antigen can be DNA, RNA, protein, peptide. The end of last year, we also introduced the usage of mRNA in order to ensure that proper expression of difficult proteins. One of them that we've had a lot of success as a difficult protein is the membrane protein qPCR. So this has been an advantage in the drug development side as well.
Perfect. Well, thank you again, Amanda, for your time today and for your important research. Before we go, I'd like to thank the audience for joining us today and for all of your interesting questions. I'd also like to thank GenScript for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until May 15th, 2025. Labroots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.